Yeah. Okay, so please start. Thanks.
Yeah, thanks for inviting me to give this BioCars seminar today, and thanks to everyone for joining, spending a time out of your busy day. We recently published this work in Science Advances, and I would like to, I'll have the acknowledgement slide, but up front that we had a very great team and they were all very helpful to get this done. And of course, that includes Irina and all the staff at BioCars to help get this work published. So the HIV-1 envelope glycoprotein, or OMV for short, mediates fusion between HIV virus and the host cell. And this protein, sorry, I'm going to turn my laser pointer on here. This protein is composed of the GP120 receptor binding domain and the GP41 fusion domain. And both the GP120 and GP41 elements undergo a series of structural changes in order to mediate fusion with the host cell. So initially, the envelope will start off in this closed structure that we refer to as state 1. And initially, upon binding the host cell receptor CD4, the envelope will move into an intermediate state 2. And eventually, it moves into its fully open structure, the state 3 structure. Ultimately, the envelope will move into the post-fusion structure, and this is where fusion is mediated. Sorry, I'm trying to switch something on my Zoom screen. Oh, well, it's going to be like that. Okay. So these movements can happen in the absence of the CD4 receptor engagement. And this is important because the envelope is the only viral protein on the surface of the HIV. And so this is our primary target for when we're designing vaccines. And controlling the envelope movements has been shown by a lot of different work in the field previously to be important for our vaccine designs. And I'll speak a little bit more to that shortly. So this image shows the linear layout of the envelope structure. And like I said, it's composed of a GP120 domain represented by this blue rectangle and the GP41 domain represented by this orange rectangle. And there are 3 subunits. So you have 3 GP120s and 3 GP41 making a trimer. So these GP120 and GP41 can be broken down into smaller structural domains that undergo rearrangements during this opening process. And for the purposes of this project, we're mostly focused with the V1, V2 elements that's shown here in green, the V3 element in red, and the beta 20, beta 21 element in yellow. So previously, cryo-EM studies have shown that there are basically 3 stable structures that we know about for the envelope. There's the closed structure that's shown here on the top, and the inset on the bottom shows the V1, V2, V3, and beta 20, beta 21 domains. Importantly to note in this structure is that at the trimer apex at the top, the V1, V2 domain interacts with the V1, V2 domain and the adjacent GP120 protomers, and these interprotomer contacts help to stabilize the closed structure of the envelope. There has also been what we call the open occluded structure, and this structure is similar to the closed structure in that the V1, V2, and V3 elements have not undergo very significant rearrangements from the closed state. But as you can see looking up here, that the GP120 protomers have actually rotated out, breaking the contacts between the V1, V2 domains and the different GP120s. Additionally, there is this fully open structure of the envelope. And in addition to the rotation of the GP120 protomers, you also see a complete rearrangement in the V1, V2, and V3 domains. They become totally disorganized in the open structure. We also see the formation of this bridging sheet here with the beta 20 beta 21 elements and part of V1, V2 as well. And so these are the structural rearrangements that happen during the course of opening that we do already know about from cryo-EM studies. So as I mentioned previously, the HIV-1 envelope is the primary, what we would call immunogen that we use for HIV vaccines. And the specific conformation of the envelope has a great impact on the type of antibodies that can be generated or how they bind or if they'll be neutralizing or not. And this is very important because there are a large variety of HIV variants, and not every antibody is capable of neutralizing a large breadth of these strains of HIV. And so what previous studies have shown is that antibodies that have the capacity to neutralize a larger breadth of HIV strains, what we would call broadly neutralizing antibodies, prefer to bind the closed structure of the envelope. And antibodies that bind to the open structure tend to be either weakly or non-neutralizing. And so this shows how the structure of the envelope is very important for the type of immune response you can elicit if you're going to be using this in your vaccine. And so because we need to understand the conformation and structure of this protein, we really do need to understand the dynamics to be able to design an effective vaccine. And I also just realized I forgot to mention something very important. So I'm going to back up to this slide for a second. The native structure of this envelope is embedded in the viral membrane. And so the structure that we work with, though, is a solubilized ectodomain construct called the SOSIP. And in this SOSIP, we have truncated out we truncate right here at this dotted line so that the ectodomain does not contain the transmembrane domain. We also introduce a disulfide between GP41 and GP120, and this proline mutation here in the GP41 peptide repeat motif. And so I just wanted to point out that this is one of the biggest structural changes, modifications we make from the native protein to stabilize it and be able to work with it, is to make it solubilized. And we call this the SOSIP construct. So yeah, sorry that I forgot to point that out first. But yeah, so then in the SOSIP constructs, these binding studies have shown that the closed structure is where the broadly neutralizing antibodies prefer the closed structure, and weakly or non-neutralizing antibodies prefer the open. And so this is why we're interested, because we think understanding the dynamics here can help us design better vaccines. So the primary goal for this project is to resolve the transition pathway for the HIV-1 envelope opening. And one of the main questions we have is the open occluded state actually a transition intermediate along the opening pathway, or is it just a structure that is captured with antibodies? We've only ever observed this structure with antibody binding. So there's still a question of if this is a state actually available to the envelope absent the antibody being bound. We also are interested in identifying other structural intermediates along this pathway and determining the kinetics associated with these transitions. So one of the things that's important to keep in mind when thinking about the envelope structural transitions is this reaction diagram, which shows the transition moving from closed to open on the x-axis and the energy on the y-axis. And this is just a sketch, so these are not real data. But the important point is that to make this transition, the protein is going to have to move through an unstable transition intermediate state. And these intermediates can be much harder to capture with traditional structural methods because they are unstable. And so, but we do believe that we need to understand what's happening in this transition like I've said before, better design our vaccines. And so we have another problem facing us when we're trying to understand HIV envelope structure, and that is that the HIV-1 envelope, the HIV-1 virus and its associated envelope actually have very high rates of mutations. So there are a large number of variants, which are kind of shown here diagrammatically. These variants have been broken down into various subgroups, with the subgroup M being the cause of most of the infections worldwide. And so the main variants we work with in the lab are the CH505 and the BG505 variants as well. We also work on adding stabilizing these proteins for vaccine design. And one of the stabilized constructs that we're going to be showing data for today is a disulfide stabilized construct called the CH848. So you'll hear me refer to the CH505 strain or BG505 variant or CH848. These are all HIV-1 envelope, either different strains or for CH505 or BG505 or different design for a vaccine for CH848. Now, in order to measure these transitions, we need a method that can measure fast transitions. In our case, we need the nanoseconds to microseconds time scale. And we also need a method that can report on short-lived intermediate structures. And we found that the temperature jump time resolved small angle X-ray scattering satisfies both of these conditions. In order to successfully apply the temperature jump method, we need an envelope variant that can readily undergo the close to open transition. They do not all readily undergo the transition the same, so we need to identify a variant that will be optimal for doing these experiments. We also need to know what the temperature sensitivity of this close to open transition is. That is not information we knew going in ahead of time, so we had to identify if the transition was temperature sensitive and what temperature range it was most temperature sensitive over. We also need to confirm that we can actually detect the differences between closed and open envelope structures with the Saxis method. So with this in mind, we asked our first question of, can we measure envelope conformational differences with Saxis? And so for these experiments, this is just static Saxis. And we need a way to correlate what we see in our Saxis profiles to some other structural element with the protein. And we chose to use antibody binding. So 17B and 19B are antibodies that specifically bind to the open state of the envelope and do not bind the closed state. So the binding of these two antibodies can be used as a proxy for the degree of openness an envelope is exhibiting. Conversely, PGT145 is an antibody that specifically binds to the closed state and does not bind to the open state. And we can use PGT145 binding as a readout for if our envelope has the proper fold. And so we measured the BLI, we measured binding by biolayer interferometry, which is BLI. And this is a method that measures the shifts in wavelengths that are associated with complex formation or dissociation. So these panels here show the BLL binding response on the y-axis for BG505 in blue and CH505 variant ONV in red. And what we can see from these plots is that the CH505 envelope has a greater propensity to occupy the open state as indicated by 17B binding on the left and 19B binding in the middle, and that it can still exhibit, and it still has the proper fold as it can still bind to PGT145 quite well. So we wanted to see if we could, if whatever structural change that is responsible for these differences in binding, we wanted to see if we could detect it with static Saxis. So we measured the static Saxis of the BG505 and CH505 variants. Again, CH505 is in red and BG505 is in blue. And we have the scattering angle on the x-axis and the scattering intensity in the log scale on the y-axis. And we can see that there is actually a noticeable feature here in this area. And this is actually a little bit easier to see when we look at the difference curve. So we subtracted the BG505 curve from the CH505 curve to get this difference curve here. And we're looking at this feature that's centered at about 0.007, or sorry, 0.07 inverse angstroms. Now, we can clearly detect some structural difference between these two with our Saxis profiles, but we still don't really have an idea of what this structural change is. So we built models for the envelope in a closed and open structure, and then calculated the theoretical Saxis curves and then the theoretical difference curves for a closed to open transition. And this is the Saxis profile we got for this theoretical close to open transition. And you can see that we're noticing the same feature at the same scattering angle value here. So from this, we interpret that we can measure differences between open and closed conformations in the envelope using Saxis. We also took this one step further and showed that and measured Saxis in a whole series of envelopes that have varying degrees of closed state stability. And we correlated this to the binding response for 17B, 19B. And 17B is shown here in blue and 19B is in red. And you can see that the correlation here is pretty tight. I mean, there's a pretty reasonable correlation. It's pretty strong for these two binding antibodies. So this is indicating to us that this is not a phenomenon that's specific to any one variant or construct of the envelope, that is a general feature that we can pick up and measure with Saxis. We also found that the closed state antibodies PGT145 and 151 did not have a correlation to the average scattering at our feature of interest, Q equals 0.07. And that is actually kind of expected because both of these antibodies actually have glycans as part of their binding site. So they're not fully just based on protein dynamics as part of their binding. So this to us makes sense. But yes, the main point here is to show that the features we're measuring are not specific to any one variant or construct design of the envelope. Now that we can confirm that we can measure open and closed structures with the Saxis, we then move to answer if we can induce opening with temperature jumps. So in these experiments, we were using the CH505 variant of the envelope because it was shown by our previous binding studies to have a higher propensity for the open state. And so we measured the binding for 19B, 17B, and PGT145 over temperatures between 20 and 50 angstroms, as you can see here, and measured the BLL binding that's shown on the y-axis. And what we found is that the env opening is indeed temperature sensitive, and it's most temperature sensitive here between 40 and 50 degrees Celsius. We also wanted to see if we could measure this same difference using static Saxis. So we measured the envelope's static Saxis signal at both 50 and 25 degrees Celsius and calculated the difference curves. And as you can see, we do identify this same feature here, even when looking at it through Saxis. And as just a reminder that this is the same feature that we saw for our theoretical close to open transition. So taken together, we say that we can indeed induce envelope opening with a temperature jump, and we can capture this opening with the Sax method. So now that we have basically been able to fulfill the assumptions and the necessary components, we were ready to move forward into our time-resolved temperature jump SAX experiments. A few things that we did that are slightly different than normal for these I wanted to point out is that, as normal, we have our sample in a aluminum nitride holder in a capillary, and it's being held at the starting temperature of 44 degrees Celsius. As typical, our capillary is going to be in line with the infrared laser pump and the X-ray detector, and of course the X-ray beam, which in this case is the pink X-ray beam. One of the things that we did in our experiments is that due to constraints of protein production, we actually had our input and output sampling through the same tube, and it was being pumped continuously using a peristaltic pump throughout the duration of the experiments. We knew that the transition was reversible and that we could recycle our protein, and we monitored for effects of radiation and damage. So I did want to point out that we were recycling and reusing our protein for the duration of these experiments. We used the infrared laser to induce the temperature jump, which we calibrated to be about 6 degrees Celsius. So our final temperature was 50 degrees Celsius. Our protein size is actually quite large. So the protein itself is about 210 kilodaltons, give or take, depending on the specific sequence you're working with. But according to mass spec studies, the protein size can get up to 300 to 400 kilodaltons with the specific glycosylation patterns on the protein. And the glycosylation patterns can be different from protein to protein and can depend on your production methods as well. So that's just another variable that we have to work with. So for these temperature jump experiments, we measured the SAX's scattering at 2 time points before hitting with the infrared laser. That would be -10 and -5 microseconds. And then we made subsequent measurements after hitting with the laser for a certain time delay. And we measured, time delays from 500 nanoseconds to 100 milliseconds in our proteins. So for our time results, this is our time resolved SAX's profiles that we were able to collect for the CH505. And this is the difference curve. So the -5 microseconds curve have been subtracted off from the time point to give these scattering profiles here. And what we notice in our scattering profiles is that at 1.5 microseconds, we can already see a scattering feature that's started, but it's not complete yet. It continues to evolve at the later time points. We can see another feature that separates out at later time points here, and another feature here. This is the one that we've been picking up in our theoretical SAX's curve. Now, this earliest time component that I first showed with the first arrow actually is happening as fast as 500 nanoseconds. This transition is basically happening faster than our dead time. So whatever it is, we're not really concerned with it for the rest of our analysis. And we're really more interested in like what's happening here and here. So at this later time point, this is the 100 millisecond curve. You can see that the scattering profiles are starting to relax back to their starting point, so that we can say that this transition is reversible, which is consistent with the prior data from the field. We were also able to measure similar profiles for the CH848 envelope as well. And one difference that I do want to point out is that we didn't actually go down to quite so low a time delay here. We started at 5 microseconds. We also have additional time points of 250 microseconds and 750 microseconds here. But what we see is that there is this similar feature here, this main one we're interested, this little lump at 0.07 inverse angstroms. So now that we had measured our time-resolved SAX's profiles, we were interested to determine what components were in those profiles. And so we used a method called Regals, which is basically an SVD analysis that is constrained by the minimum and maximum dimensions of the protein. And we performed this Regal analysis on the CH505 time-resolved axis difference curves that I showed previously. And what we found was that there were two primary components in the data, shown here as component 1 in blue and component 2 in red. We can see that component 1 kind of looks like the feature that is like shooting down in the previous difference curves, but inverted. And then component 2 looks very similar to the feature that we observe at the 0.07 inverse angstroms. We also see a similar components for the CH848 envelope, and this is somewhat surprising because the CH848 is supposed to be stabilized in the closed state. It's not really supposed to be going through any of these opening transitions, but we can see here that it is going through, at least theoretically, some similar transitions. So that was interesting. But we still have more work to do to identify what these components are and if they're real. But first, we wanted to know how fast what kinetics we can associate with these components. So we also ran a standard SVD analysis, and the left vectors from that SVD analysis matched the components we got from the Regals analysis. But in this case, to determine kinetics, we were interested in fitting the right vectors. So for the first right vector, we fit a double exponential equation model, and then for the second, we fit a single exponential model. And from these fits, we found that there was a fast motion and a slow motion. And the single exponential fit to the second component had a value consistent with this slower component here. We also fit the CH848 data using the same models, a double exponential for the first component and a single exponential for the second component. And we found, again, this was shocking, that these transitions are happening and they're not slower. We thought they would at least be slower, but they were not slower, and they might even be faster. So that was very unexpected. Now, one of the big questions, of course, is like, we've measured this great data, we found these components, got kinetics to go with it, but what are the actual transitions? What atomic models can we associate with this data? Again, I go back to my Regals analysis, because the one thing nice about the Regals analysis is it outputs a pair distance distribution for each component. Since we use difference curves for our Regals calculation, these are pair distance distribution differences. So they're showing us the changes in the different distances that we're seeing. So for the CH505 variant of envelope, we see that there is, okay, this is a little bit confusing, but because of how their difference curves are calculated, this increase, this little positive value here, is actually indicating that there is a, wait, sorry, I gotta think this, that there is actually a decrease in the, that there's actually a decrease in the, sorry, I'm saying this one. Anyways, there's a change in these components here at 50 angst, centered at around 50 to 60 angstroms. And we also see changes going from 50 to about 175 angstroms. See the same thing for CH848. But we still don't know what the atomic models are. We built atomic models for envelope in a lot of different conformations and calculated theoretical axis curves and pair distance distribution curves for theoretical transitions, and try to match these to what we have from our experimental data. We built models for a closed to open occluded transition, a closed to open transition, an open occluded to open transition. And the difference curves, which are shown below the cartoons on the left-hand side, and the pair distance distributions, which are on the right-hand side, don't match what we see in our experimental data, so that these these are not transitions that are contributing to our sexist components. However, both the difference curve and the pair distance distribution matches for our modeling. So, or what we get from our experimental data. So this indicates to us that our slower transition, component 2, is associated with the rigid body rotation of the GP120 domains. We had a harder time figuring out what component 1 was. We suspected it maybe could be some rearrangements in the V1, V2, or V3 domains. So we ran 250 replicas of molecular dynamics simulations, each for 5 microseconds on a glycosylated GP120, which you see right here. And what you can see from watching these simulations is that while there is plenty of dynamics in the glycans and even quite a bit in the V1 domain, especially right here in this loop, we're not seeing a movement into an open structure where these V1, V2, and or V3 is fully released. So this indicated to us that the V1, V2 domains are not being released and are not contributing to our sexist signal, but we did perform a root mean square deviation analysis over the aggregate simulation. So this is an aggregate time of 1.25 milliseconds. It's a simulation time. The dotted line in these histograms of the RMSD shows what the RMSD value would be for the open structure. And what you can see from these plots is that the V1, V2, and the V3 domains do not release, and they do not move or even begin to transition into an open structure. So we interpreted this as that these motions are not contributing to the sexist signal in the 5 to 10 microseconds time scale. The pair distance distributions that Regal showed us were actually consistent with the disruption of the apex context. So we actually built a model for a transition going from a closed structure to a closed structure where those contexts at the apex are disrupted, and calculated the theoretical difference and pair distance distribution difference for this. And we found that this actually matches what we see for component 1 in our Regal's data. And so we associate the fast component 1 transition with an order to disorder transition at the envelope trimer apex. And so to summarize, like where we started and what we found, coming into this work, we knew about the closed structure, and we have a particular interest in learning how to stabilize the envelope in this structure. And through previous cryo-EM structures, we knew that there was an open occluded structure, but and assumed it was an intermediate on the way to the open structure, but we're not sure about this. And we're not sure if this is the pathway or what pathway there is. And we set out to explore some of the transition intermediates for this pathway. And what we found is that the movement from the closed to open occluded structure is happening on approximately 500 microsecond timescale. And this is consistent with previous data, FRET data in the field, showing that there is a transition happening faster than the millisecond timescale. We also were able to identify in this closed to open occluded transition a new microsecond intermediate that has not been identified before, and that this intermediate forms very quickly, about 10 microseconds into the opening process. We weren't able to measure all the way to the open structure, but binding studies indicate that this could take on the seconds to hours time scale, but probably more work would need to be done to pin that down. And also, I just want to point out and make sure everybody understands that these rates are definitely dependent on the specific envelope sequencer that we're using here. So these are pulled mainly from the CH505 data. The values might be slightly different for CH848. Now, in the Henderson lab, we're not only interested in the basic biology of how the envelope functions and works, but we're interested in leveraging this information to design vaccines. So the last question we have is, how can we better stabilize this closed envelope? The vaccination studies have shown us that envelopes that we thought was stable, and you saw from the Saxis data that envelopes we thought were stabilized in the closed state actually have the ability to move into the open occluded state. And we got to thinking that this might be because of that initial fast 10 microsecond transition where the apex contacts just break apart. So we sought to design, using a disulfide staple, a model that would lock the GP120 contacts closed so we can prevent the movement into the open occluded structure. So as I said, we engineered a disulfide staple between the adjacent GP120s, and from this cryo-EM structure, you can see that we were able to successfully introduce that disulfide into the envelope. We did some binding studies and found something quite interesting and shocking, is that this staple design was actually able to bind an antibody called B12. And this was shocking to us because B12 is an antibody that has been shown to bind the open occluded structure and not bind the closed structure. We did not believe it was able to bind the closed structure, but we saw in our cryo-EM studies and through our binding studies on this design that it indeed does bind, and it is in a closed structure. And you can see that from looking at these two diagrams on the bottom, that the way the structure of the envelope is different. Like I said, the contacts are disrupted at the trimer apex, but the mode of binding of the antibody is also very different as well. So we looked at this comparison a little more closely. And we look in the GP120 overlays. So you see the disulfide staple in blue and the open occluded structure here in green. And there's not that significant of differences in this GP120 structure here. But when we look at the antibodies with the closed structure in this orange color and the open occluded bound B12 in the red, we can see that there's actually a pretty significant degree shift in the angle of approach for this antibody binding to the GP120. And so this was very interesting to us. It indicates that the conformation and the stabilization efforts we make to stabilize the closed state, they have significant impacts on the antibody binding and the basically immune response you can generate if you put one of these into a vaccine. So the importance of the work that we've done is that, well, we were able to identify that the early structural rearrangements in HIV and SOCIP involve at least 2 rapid transitions. And the modeling suggests that the fast transition involves a loss of trimer apex contacts, while the slow transition is consistent with rigid body contacts of the GP120 protomers. And surprisingly, the initial fast transition appears to be somewhat faster in the stabilized CH848 envelope construct. We also showed with this work that the GP120 interprotomer disulfides can stabilize the closed envelope, and that the stabilization of these interprotomer contacts can significantly influence your antibody binding. And so with that, I can take any questions.